we are going to talk about the engine of the internal organs, the tissue that actually keeps you alive while you sleep, while you digest, while your blood circulates. We are diving deep into smooth muscle. And I want to be clear right off the bat. This isn't going to be a high-level, oh, it works automatically kind of chat.
No. The mission today is granular. We are going to explain every single mechanism we can. We want to understand the physics, the chemistry, and the electricity that allows your bladder to hold urine for 6 hours without you getting tired.
Or your blood vessels to regulate pressure for 80 years. It's just amazing.
It is. We want to get into the nuts and bolts of it all.
It's surprisingly different from skeletal muscle, isn't it?
Yeah.
I think most people, myself included before this, just assume muscle is muscle. You flex it, it shortens. Same deal.
Right.
But reading through the source material, smooth muscle feels like it was designed by a completely different engineer on a different planet almost.
It really does. If skeletal muscle is, say, a high-performance sports car, built for speed, for power, for quick bursts, then smooth muscle is a heavy-duty industrial generator.
I like that analogy.
Yeah, it's designed for one thing above all else: efficiency and endurance. It operates on different rules, different timelines, and it uses a completely different chemical trigger to get started.
So let's start with the name itself. Smooth muscle. Why smooth? Is it just a texture thing?
It's a visual description, fundamentally. If you put skeletal muscle under a microscope, it looks striated. It has these very clear stripes, light and dark bands.
Right, you see those diagrams in every textbook.
Exactly. And that's because the contractile fibers, the actin and myosin, are arranged in these perfect, rigid, repeating regiments. It's incredibly organized. Smooth muscle.
Well, it lacks that striation.
So under the scope, it just looks...
It looks smooth, uniform,
Right.
but, and this is a key point, that lack of stripes doesn't mean it's disorganized. It just means the organization is different, more of a three-dimensional web than a two-dimensional set of rows.
Okay, so that's the smooth. Let's get into the physical dimensions first, the
Yeah.
anatomy of this automaton. We are talking about fibers that are significantly smaller than the skeletal muscles we're used to.
Oh, tiny by comparison, just microscopic. Let's look at the data points.
Yeah.
The typical smooth muscle fiber is usually only about 1 to 5 micrometers in diameter,
Right.
and its length can vary, but maybe 20 to 500 micrometers long. Okay, 1 to 5 micrometers in diameter. Help me contextualize that. How big is a human hair?
Well, human hair is what, maybe 50 to 70 micrometers? So
Yeah.
you could line up 10 or 20 of these smooth muscle fibers side by side to match the width of one hair.
Wow. Okay, and compared to skeletal muscle?
Well, a skeletal muscle fiber is a monster. It can be 30 times wider in diameter and hundreds, sometimes thousands of times longer. So smooth muscle fibers are these discrete microscopic spindles. But the magic isn't in the individual fiber, it's in how they are arranged.
Right. And this brings us to the first major fork in the road for our discussion. The two major types of smooth muscle.
Yes, this is a critical distinction.
Right, I saw this in the breakdown. Multi-unit versus unitary. And the names are a little counterintuitive at first glance, especially unitary.
They are. Let's unravel them. First up, multi-unit smooth muscle. The key word here is independence. This tissue is made up of discrete, separate fibers. Each one operates pretty much independently of its neighbor.
So they aren't chatting with each other? There's no teamwork?
No. In fact, they're actively insulated from one another. Each fiber is covered by its own little sheath, a basement membrane made of collagen and glycoprotein that electrically isolates it.
And the control system for them is also very specific.
Extremely specific, and this is the other crucial part. Each fiber is usually innervated by a single nerve ending. It's a one-to-one connection, much like we see in skeletal muscle.
One nerve, one fiber that just screams precision control.
That is exactly the point. Precision. Think about the places you find this stuff. The ciliary muscle in your eye that controls the focus of your lens.
Which needs to make tiny rabbit adjustments.
Exactly. Or the iris, which controls your pupil size, opening and closing with minute changes in light. You don't want a big, clumsy wave of contraction there. You want exact, graded adjustments.
And the polorector muscles, the little guys that give you goosebumps, that's them too.
That's them. A perfect example. When you get goosebumps, that is multi-unit smooth muscle. It's a specific neural event triggering specific individual hairs to stand on end. Precision.
Okay, that makes sense. One-to-one control for fine tasks. But then we have the second type, the one that runs the major organs, unitary smooth muscle.
Right. And this is where the name unitary trips people up. It sounds like it means one fiber, but it actually means the exact opposite. It means it acts as one unit.
A single coordinated unit.
Correct. You'll also hear it called syncytial smooth muscle, or more commonly, visceral smooth muscle, because it's found in the viscera, the gut, the bile ducts, the ureters, the uterus, the walls of most blood vessels.
So in this case, you have hundreds or thousands of fibers all bundled together.
Bundled together in sheets or layers, yes. And they are not insulated like the multi-unit ones.
So they're definitely talking to each other.
They're physically and electrically connected. Their cell membranes actually adhere to each other at multiple points, which allows physical force to be transmitted directly from one fiber to the next. But more importantly, they are connected by what are called gap junctions.
Gap junctions. I love that term. It sounds like a portal from a sci-fi movie.
That's effectively what it is. It's not just a fancy name. A gap junction is a low-resistance pathway between cells. It's literally a channel, a protein portal, that allows ions, the charged particles that create electricity, to flow freely from the inside of one cell directly to the inside of the next.
So if one cell gets an electrical spark, an action potential, it doesn't stay contained within that cell.
Not at all. It flows instantly, like a current through a wire, to the next cell and the next and the next. The action potential travels rapidly over the entire sheet of muscle.
And that's why your stomach doesn't just twitch randomly in a million different spots.
Exactly. It contracts in a coordinated, powerful wave. The whole tissue acts as a single functional unit, hence unitary.
That distinction is huge.
Yeah.
It's the difference between an orchestra where every musician has their own sheet music and conductor versus a choir where everyone sings the same note at the same time when the signal is given. Precision versus mass action.
That's a great way to put it.
Okay, so we have the layout. Multi-unit for precision, unitary for coordinated squeezing. Now let's look at the engine itself, the actual mechanics of contraction. In skeletal muscle, we always talk about actin and myosin, the sliding filament theory. Do we have that here?
We do. The fundamental chemical basis for contraction is still the interaction between actin and myosin filaments. The physics of attraction, that pulling action, is the same. But the architecture, how those filaments are hung within the cell, is radically different. And there's one massive glaring omission.
The missing player.
The troponin complex. This is so important. In skeletal muscle, troponin is the master regulator. It's the lock on the door that sits on the actin filament and physically prevents the myosin from binding.
Until the key arrives.
Until the key calcium arrives and binds to troponin, causing it to change shape and move out of the way. Smooth muscle does not have the troponin complex.
Which implies that the entire control mechanism, the thing that says go or stop, must be totally different.
Completely different. And we will get to that chemistry because it's fascinatingly complex. But first, let's visualize the structure. If there are no stripes, no rigid Z discs to anchor the actin filaments, what is everything holding on to? They are anchored to structures called dense bodies.
Dense bodies.
Yes. Imagine a kind of three-dimensional web-like scaffolding inside the cell. The dense bodies are the anchor points, the nodes of this web. Some of them are attached directly to the inside of the cell membrane, and others are just sort of floating inside the cell, suspended by structural proteins.
And these serve the same function as the Z discs in skeletal muscle.
Functionally, yes. They are the anchor points for the actin filaments. So you have these thin actin filaments radiating out from the dense bodies, almost like spokes from the hub of a wheel.
And the myosin is in between.
The thicker myosin filaments are interspersed among the actin filaments, positioned to interact with them. And there's another level of connection. The membrane-dense bodies of adjacent cells are often bonded together by intercellular protein bridges.
So the force can be transmitted not just electrically, but physically from cell to cell.
Exactly. The whole tissue is physically linked together, which contributes to that unitary contraction.
It sounds chaotic, though, compared to the neat rows of a skeletal muscle sarcomere.
It looks chaotic, but this apparent chaos allows for something that the rigid structure of skeletal muscle can't do. Skeletal muscle is built to pull in a straight line very powerfully. Smooth muscle, with this web-like structure, can contract in 3 dimensions, kind of balling up on itself, and it does this using a side-polar cross-bridge arrangement.
Okay, this was the part of the research that really made me pause and reread it a few times, the side-polar twist. Break that down for us.
Okay, let's visualize a single myosin filament. In skeletal muscle, the little heads that stick out, the cross bridges, are arranged to pull everything towards the center of the filament. It's a symmetrical pull inward.
Right, like 2 teams in a tug-of-war pulling on the same rope.
A good analogy. But in smooth muscle, the myosin is hinged differently. The bridges on one side of the filament are hinged to pull in one direction, and the bridges on the other side of the filament are hinged to pull in the opposite direction.
So it's pulling two different things in opposite directions at the very same time.
Exactly. One myosin filament is essentially pulling one actin filament to the left and another actin filament to the right simultaneously. It's like a two-way winch.
Wow. Okay. And what is the functional consequence of that strange arrangement?
The result is staggering. Because of this, and because there are no rigid Z-discs blocking the movement at the end of a sarcomere, smooth muscle cells can contract by as much as 80% of their length.
80%.
Yes,
Yeah.
80. For comparison, skeletal muscle is limited to shortening by less than 30%. It hits a physical wall. Smooth muscle just keeps going.
That is a staggering difference. Why do we need 80% compression in an organ?
Think about the hollow organs, the stomach, the bladder, the uterus. Let's stick with the bladder. It needs to expand to hold a significant volume of liquid, maybe half a liter or more, and then it needs to contract all the way down to almost zero volume to completely empty itself.
If it could only contract 30%, you'd never be able to fully empty your bladder.
Never. You'd be walking around with it mostly full all the time, which would be problematic. This 80% contractile ability is essential for any organ that has to function as a variable volume container.
That makes perfect sense. It's a volume management issue. The structure is built for the function.
Always. Form follows function, so the physical structure is designed for these extreme changes in size. But the other side of the coin, the other design principle, is economy. You mentioned this in the intro. Smooth muscle is the ultimate energy saver.
It's the Prius of the muscle world.
It's even better than a Prius. It's unbelievably efficient, but the trade-off is that it's remarkably slow. Let's talk about the cycling speed of those cross bridges.
Okay, the rate at which the myosin heads attach, pull, and then release from the actin.
Exactly. That frequency is very, very low in smooth muscle. We're talking 1/110th to as slow as 1300th the speed of a fast skeletal muscle fiber.
1/300th. That is glacial. Why is it so slow? What's the mechanism?
It comes down to enzyme activity in the myosin head itself. The myosin head has what's called ATPase activity. ATPase is the enzyme that splits an ATP molecule to release the energy for the power stroke, the pull. In smooth muscle, this ATPase activity is far, far lower.
So the chemical reaction that powers the movement is just happening in slow motion.
In extreme slow motion. And because the energy release is slow, the whole cycle slows down.
But paradoxically, this saves energy. It seems like being slow would be inefficient.
Massively. And here's why. Because the cycle is so slow, the cross bridge stays attached to the actin for a much longer period of time during each pull. And remember, one complete cycle attach, pull, detach uses one molecule of ATP.
Right.
So if the cycle takes 10 times longer, you are getting 10 times the holding duration or tension time for the exact same energy cost.
So it's not burning fuel just to stay still and hold a position.
Precisely. This means smooth muscle requires only 1/110th to 1300th as much energy to maintain a given amount of tension compared to skeletal muscle. This is absolutely crucial for organs that need to maintain tonic contraction.
And what's tonic contraction?
Just a constant state of squeeze. Think about your blood vessels, which are always slightly constricted to maintain blood pressure, or the sphincters in your digestive tract. They have to stay closed for hours. If they burn energy like your biceps do when you hold a weight, you'd be metabolically exhausted just by, maintaining your blood pressure.
That explains the incredible energy efficiency. But does this slowness apply to the startup time as well? Is it sluggish to get going?
It is very sluggish. After a signal arrives, a typical smooth muscle tissue takes 50 to 100 milliseconds just to start contracting. Then it can take half a second or more to reach its full force. And relaxing takes even longer, maybe 1 or 2 seconds to decline.
So the entire event from start to finish can be 1 to 3 seconds long.
Easily. And that's about 30 times longer than a single skeletal muscle twitch. And this varies widely, by the way. Some smooth muscle can take up to 30 seconds for a single contraction relaxation cycle.
So you definitely don't want smooth muscle for your hamstrings. You'd never win a race.
No, you would fall over. But you absolutely want it for your gut. You want slow, sustained, rhythmical squeezing to move food along, not a violent, twitchy mess.
Okay, now here's the fact that I found the hardest to believe when I was reading the sources. It seems like a total contradiction. Despite being smaller, despite having fewer myosin filaments, and despite being incredibly slow, The source material says the maximum force of smooth muscle per square centimeter is actually greater than skeletal muscle.
It's true. It's one of the most surprising things about it. Smooth muscle can generate 4 to 6 kilograms of force per square centimeter of cross-sectional area. Skeletal muscle is usually in the range of 3 to 4.
How is the weaker, slower, smaller muscle stronger? That makes no sense.
It goes right back to that slow cycling and the prolonged attachment time. Because the myosin heads stay attached for so long, at any given microsecond, a much larger percentage of the total heads are attached and actively pulling.
Ah, okay. So in fast skeletal muscle, a lot of the heads are in the reset phase at any given moment. They're detached and getting ready for the next pull.
Exactly. There's a lot of cycling, but not all of them are pulling at once. In smooth muscle, they're not cycling quickly. They are latching on and holding. The cumulative force of all those simultaneously attached heads is what generates that immense sustained power.
Strength through persistence, not speed.
Precisely. And this leads us directly to another unique and incredibly important feature, the latch mechanism. This is a specific physiological state where the muscle acts almost like a clamp or a ratchet.
Explain the latch mechanism. This seems to be key for that whole autonomy and economy aspect we've been talking about.
It's central to it. So imagine a blood vessel has constricted. Once it has achieved that full contraction, the amount of excitation, the nerve signal, the calcium levels can drop significantly, way down to a trickle.
Yeah.
But the muscle doesn't relax. It maintains that full tension.
It's like engaging a parking brake. You press the pedal hard, set the brake, and then you can take your foot off.
That's a very similar idea. It holds the tension with very little ongoing input. The theory, which we can get into the chemistry of later, is that the enzymes responsible for detaching the myosin head get deactivated while the head is still attached to actin, so the head gets stuck. It's latched in the pulling position.
And if it's stuck, it's not cycling.
Oh.
And if it's not cycling,
it's not using ATP. The energy cost of maintaining this latch state drops to almost nothing. This is what allows your vascular smooth muscle to stay clamped down to maintain blood pressure 24/7 with minimal metabolic cost. It's an incredible feat of biological engineering.
that is brilliant. Truly brilliant.
It really is. And there's another related property also vital for hollow organs called stress relaxation. This brings us back to the bladder scenario.
Right. Walk us through what's happening when the bladder is filling up.
Okay, so your bladder is slowly filling with urine from the kidneys. The volume is increasing, which means the muscle wall is being stretched. Initially, that stretch causes an immediate reflexive contraction and a spike in pressure. If you plotted it on a graph, the pressure inside the bladder shoots up.
That's the initial "I need to go" signal you feel.
Right, that's your nervous system sensing the pressure. But then something amazing happens. Within about 15 seconds to a minute, despite the volume staying high or even continuing to increase, the pressure drops back down almost to its original level.
The urge passes.
The urge passes. This is stress relaxation. The muscle fibers are physically rearranging themselves, lengthening to accommodate the new, larger volume without fighting back. It allows the bladder to continue filling while keeping the internal pressure low and stable.
And it works in reverse, too. The sources mentioned reverse stress relaxation.
Yes. When you empty the bladder, the volume suddenly drops to 0. This causes an immediate drop in pressure. But then, over the next few seconds, the muscle tightens back up, increasing the pressure back to its baseline tone. It ensures the organ is never just a flabby, empty sac. It always has some inherent tone ready for the next filling cycle.
It's an adaptive container.
Incredible. Okay, we've covered the structure and the mechanics. Now we have to do the heavy lifting, the chemistry. We established early on, and this is the big one, there is no troponin. So how does this thing turn on? Skeletal muscle is simple. Calcium hits troponin, boom, contraction. What is the Rube Goldberg machine for smooth muscle?
It's definitely a more complex, multi-step process.
Yeah.
But it still starts with the same universal trigger.
Calcium.
Calcium. It all begins with an increase in intracellular calcium ions.
Okay, so step 1.
Step 1. Calcium concentration increases in the cytosolic fluid. We'll talk about where that calcium comes from in a minute because there's another huge difference. But for now, just know that calcium levels inside the cell go up.
Calcium enters the chat. Okay.
Yeah.
Step 2. This free calcium doesn't have troponin to bind to. Instead, it binds reversibly with a different regulatory protein called calmodulin.
Calmodulin. So this is the functional replacement for troponin. It's the protein that catches the calcium.
Functionally, yes. It's the initial calcium sensor. Now you have a calcium calmodulin complex.
Okay, so they're a little team now. What do they do?
Step 3. This calcium-calmodulin complex goes and finds an enzyme that's just floating around, an enzyme called myosin light chain kinase, or MLCK for short.
MLCK. Okay, and I remember from biochemistry, kinase always means it's adding a phosphate group to something.
You got it. It's a phosphorylating enzyme, and that is step 4. The activated MLCK takes a phosphate group from an ATP molecule and attaches it to a specific part of the myosin head called the regulatory light chain.
It tags the myosin head with a phosphate.
It turns the key, it activates it, and that's step 5. Once that myosin head is phosphorylated, and only then does it gain the ability to bind with actin and start the cross-bridge cycling. Contraction begins.
This is a really crucial difference. In skeletal muscle, the myosin is always ready to go. It's just physically blocked by troponin. In smooth muscle, the myosin itself is dormant. It's switched off until this entire chemical cascade phosphorylates it and wakes it up.
Exactly. It's a chemically activated motor, not a physically unblocked motor. A very important distinction.
Okay, so that's how we turn it on. We're contracting. How do we turn it off? In skeletal muscle, you just pump the calcium away and it stops.
Here, just removing the calcium isn't enough to stop it. Yes, you absolutely have to get the calcium levels back down. The cell has pumps that actively move calcium out into the extracellular fluid or back into its internal storage, the sarcoplasmic reticulum. That's the prerequisite for relaxation.
But the myosin head is still phosphorylated. It still has that tag on it.
Right. So even if all the calcium leaves and the MLCK enzyme turns off, the tag is still there, and the myosin head remains active. It will keep cycling.
So you need another enzyme to come and remove the tag.
You need the cleanup crew. This is another enzyme called myosin phosphatase, or more specifically, myosin light chain phosphatase.
Phosphatase, which means it removes the phosphate, the opposite of a kinase.
Precisely. It finds the phosphorylated myosin head and it chemically splits that phosphate group off. Then, and only then, does the cycling stop and the muscle relax.
So the state of the muscle, whether it's contracting or relaxed, is really a constant battle, a tug of war between these two enzymes. The kinase trying to add phosphates and turn it on, and the phosphatase trying to remove them and turn it off.
That is the perfect way to visualize it. The level of contraction is determined by the relative balance of activity between the kinase and the phosphatase. And the relaxation time depends heavily on how much active phosphatase is present in the cell to do that cleanup work.
And this brings us back to the latch mechanism theory. How does this more complex chemistry explain the latch state?
Well, it fits perfectly. Imagine a scenario where both the kinase, the activator, and the phosphatase, the deactivator, are both highly active. The cycling is fast, the heads are being phosphorylated and dephosphorylated quickly.
Okay.
But what happens if the initial excitation signal drops a bit? The calcium levels go down, so the kinase activity slows way down. But what if, for some reason, the phosphatase activity also slows down or is inhibited?
Then the myosin head gets stuck with a tag on. It's been phosphorylated, but the enzyme that's supposed to remove the phosphate is slow to act.
Exactly. It gets stuck in the attached phosphorylated state. It's holding tension, but the enzymes needed to complete the cycle, specifically to detach and reset it, are inactive or very slow, so it just holds. That is the chemical basis of the latch. It maintains tension with very little ATP use because there's no cycling happening.
Fascinating. It's a system designed to get stuck on purpose. Now, I want to talk about the fuel source for this, or rather the trigger source. You mentioned in step 1 that calcium enters the fluid. In skeletal muscle, we know that all the calcium is stored inside the muscle in a specialized network called the sarcoplasmic reticulum, or SR. It's a self-contained system.
Right. Skeletal muscle basically brings its own lunch to work. It has a huge, well-developed SR that's packed with calcium, ready to be released instantly. Smooth muscle, well, it relies on takeout.
The notes describe the SR in smooth muscle as slightly developed or even rudimentary.
It is. In many types of smooth muscle, it's very small and not nearly as organized. It's there, but it's not the main player. In most visceral smooth muscle, the vast majority of the calcium required for contraction actually enters from the outside, from the extracellular fluid, ECF, at the moment of excitation.
So when the cell gets an electrical signal, it just opens calcium channels on its membrane and lets calcium flood in from the blood and the surrounding tissue fluid.
Yes, and this has a major implication for timing. Getting that calcium from the outside of the cell membrane diffused all the way into the center of the cell where the actin and myosin are,
Huh.
that takes time.
How much time?
It can take about 200 to 300 milliseconds, and that delay is the latent period we talked about earlier.
Correct. That's why the contraction is so slow to start. It's literally waiting for the delivery driver to show up with the calcium.
That's a great analogy for it.
What about these caveolae? The notes mention them as being rudimentary analogs to the T-tubules of skeletal muscle.
Right. So T-tubules in skeletal muscle are deep invaginations that carry the electrical signal into the core of the fiber. Smooth muscle doesn't have those. Instead, it has these little dimples or pouch-like invaginations of the cell membrane. Those are the caveolae.
And they have a purpose.
Yes, they increase the surface area, and they often lie very close to the rudimentary SR tubules that are inside the cell. So the theory is that when an action potential excites the caveolae, the calcium entering there can trigger the SR to release its own small stash of calcium. It's an attempt to speed things up a little.
A little signal boost.
Mm-hmm.
So the more extensive the SR, the faster the muscle can contract.
As a general rule of thumb, yes. But the reliance on extracellular fluid is the main headline here. And there's a critical so what for the listener in all of this.
Which is?
Because smooth muscle is so heavily reliant on calcium from the outside, its force of contraction is highly, highly dependent on the concentration of calcium in your extracellular fluid.
In your blood, essentially, your blood calcium level.
It's exactly. If your extracellular calcium concentration drops drastically for some reason, smooth muscle contraction can weaken or even stop completely. The gut stops moving, the blood vessels lose their tone. Skeletal muscle is barely affected because it has its own private internal stash. But smooth muscle is at the mercy of your moment-to-moment blood chemistry.
That is wild. Your hydration and your electrolyte balance are literally powering the engine of your internal organs in real time.
It puts a whole new perspective on nutrition and why those minerals are so important, doesn't it?
It really does. Okay, let's shift gears to control. Nervous and hormonal control. We talked about how multi-unit muscle has those specific one-to-one nerve endings. But for unitary muscle, the big sheets in the gut, the vessels, it's not so neat and tidy.
No, not at all. In skeletal muscle, you have the neuromuscular junction, a highly structured, specific synapse between the nerve and the muscle fiber. In smooth muscle, the autonomic nerve fibers just sort of branch diffusely on top of the muscle sheet. We call them diffuse junctions.
It's like crop dusting. The nerve just flies over and sprays the chemical.
That's a really good analogy, actually. The nerve fibers don't even make direct contact with the muscle cells in many cases. The fine terminal axons have these things called varicosities.
Varicosities. Describe what those look like.
They look like beads on a string. Along the final length of the axon, there are these little bulbous swellings. These varicosities are packed with vesicles containing the neurotransmitter. As the signal travels down the nerve, these varicosities just release the chemical into the general vicinity, into the matrix coating the muscle. It then has to diffuse through the fluid to reach the cells.
So it's a chemical cloud that just drifts down onto the muscle sheet.
Effectively. And because of the gap junctions we talked about earlier, you don't need to hit every single cell. You just need to excite the cells on the outer layer, and the electrical signal will propagate inward through the whole mass.
Okay, now the chemicals themselves, we usually think acetylcholine makes muscle move.
In skeletal muscle, that's true. It's always acetylcholine, and it's always excitatory. But in the autonomic nervous system controlling smooth muscle, we have two main players, acetylcholine and norepinephrine. And here's the paradox. They are never secreted by the same nerve fibers, and they usually have opposite effects on a given organ.
Give us an example of that.
Okay, in the gut, acetylcholine is generally excitatory. It causes the smooth muscle to contract and increases peristalsis. But norepinephrine is inhibitory. It causes the gut to relax.
But what about in the blood vessels?
In most blood vessels, norepinephrine is the primary excitatory neurotransmitter. It causes vasoconstriction, it makes them contract.
So how does the muscle know what to do if the same chemical, norepinephrine, means go in one place and stop in another?
It's not about the chemical key, it's all about the lock it fits into. This is the receptor rule. Whether a neurotransmitter excites or inhibits a smooth muscle cell depends entirely on the type of receptor protein on the surface of that cell.
So if a cell has an excitatory receptor,
then when the neurotransmitter binds to it, that receptor will open sodium or calcium channels. Positive ions will flow in, causing depolarization and leading to contraction.
and if it has an inhibitory receptor,
Then the binding might do the opposite. It might close those sodium and calcium channels, making it harder to excite, or more commonly, it will open potassium channels.
And opening potassium channels does what to the cell?
Potassium is positively charged, and its concentration is high inside the cell. So when you open potassium channels, it flows out, taking its positive charge with it. This makes the inside of the cell more negative. We call this hyperpolarization.
It pushes it further away from the firing threshold?
Exactly. It makes the cell harder to fire. It essentially puts the muscle to sleep.
So the receptor determines the reality for that cell. That is a crucial concept.
Absolutely fundamental. The neurotransmitter is just a message. The receptor is the interpretation.
Okay, let's talk about the electricity itself, the action potential. We know that skeletal muscle has a very negative resting potential, around negative 90 millivolts. It's in a deep freeze, hard to fire accidentally.
Yeah.
What about smooth muscle?
It lives a bit closer to the edge. Its resting membrane potential is typically less negative, usually somewhere between negative 50 to negative 60 millivolts.
And its action potentials come in a couple of different flavors. I'm looking at figure 8 to 7 in the notes here. We have spike potentials, and then we have action potentials with plateaus.
Right. The spike potential is the classic one we see in nerves. It's a very rapid, sharp spike of depolarization, maybe lasting only 10 to 50 milliseconds. You see this in unitary smooth muscle when it's stimulated by nerves, hormones, or even by stretch.
But the plateau, that's the weird one. It looks totally different.
The plateau is absolutely essential for the function of certain organs like the ureter, the uterus, some vascular smooth muscle. The potential shoots up fast, just like a spike, but instead of repolarizing and coming right back down, it stays depolarized. It plateaus for a long time, sometimes up to 1,000 milliseconds. That's a full second.
Why do you need to hold the cell in an electrically excited state for a full second?
Because it supports prolonged contraction. Think about the ureter squeezing a wave of urine down to the bladder, or the uterus contracting powerfully during labor. You don't want a quick twitch. You want a sustained, powerful squeeze that lasts a second or more. The electrical plateau holds the contraction on for that entire duration.
And what is the ionic basis for that plateau? What causes it?
It's the calcium channels again. This is another key difference. Smooth muscle cell membranes have very few fast voltage-gated sodium channels, which are responsible for the fast spike in nerves and skeletal muscle. Instead, they have a huge number of voltage-gated calcium channels.
And those are slower to open and close.
Much slower to open and very slow to close. That slow opening contributes to the slower onset of the action potential, and the very slow closing is what maintains the plateau. It keeps letting positive calcium ions trickle in, holding the membrane potential up.
And here's the double duty aspect again, right? The
Yep.
calcium that is flowing in to create the electricity.
Is the very same calcium that is then diffusing to the contractile filaments to trigger the contraction via the calmodulin pathway.
That is just beautifully efficient. The electrical signal is the chemical activator.
One and the same. In skeletal muscle, the electrical signal is a separate event that causes a secondary release of calcium from the SR. Here, the ion creating the electricity is also the trigger.
Now, what about slow wave potentials? The notes call them pacemaker waves. This is another electrical phenomenon.
This is particularly important in the unitary smooth muscle of the gut. If you were to hook an electrode to the gut wall, even when it's resting, you would see a slow, undulating, rhythmic waving of the voltage. These are not true action potentials. They are sub-threshold waves.
What causes them? Is it nerves?
It's believed to be an intrinsic property of the smooth muscle cells themselves, perhaps a waxing and waning in the activity of ion pumps, like the sodium-potassium pump. But their function is to set the rhythm. They are the metronome of the gut.
So how do they lead to a contraction?
They don't always. But when the peak of one of these slow waves rises high enough to hit the firing threshold, which is about Naxal 35 millivolts, it triggers a burst of one or more true action potentials that ride on top of the wave. And that causes a rhythmic contraction.
So the slow wave decides when contraction is allowed to happen.
Exactly. This is why your digestion is rhythmic and not just one continuous squeeze. It's paced by these intrinsic electrical waves.
And you mentioned earlier that stretch can trigger this system too.
Yes, and this is the basis of automatic peristalsis. When you fill the gut with food, the physical stretch of the muscle wall makes the membrane potential less negative. It pushes it closer to the threshold.
So it makes it easier for those slow waves to reach the firing point.
Precisely. So the sequence is, food enters up, stretches the gut wall, lowers the resting membrane potential. The peaks of the slow waves now cross the threshold. This triggers bursts of action potentials, and that causes a powerful rhythmic contraction to move the food along.
All done automatically. The gut senses the food and moves it. all via physics and electricity.
A self-regulating system.
Okay, we are entering the final stretch of the mechanisms here. There's a section on contraction without action potentials. We talked about how some of those small multi-unit fibers are too tiny for a big spark.
Right. In places like the iris or the tiny piloerector muscles, the fibers are so small they can't really propagate a full-blown action potential. It's not necessary. In those cases, the nerve releases the neurotransmitter. It causes a local depolarization right under the nerve ending. We call this a junctional potential. And that small electrical change spreads electrotonically over the short fiber.
So it's not a self-regenerating wave, just a local change that's enough to do the job.
A quiet activation. No big spike, just enough depolarization to open some calcium channels and cause contraction.
And finally, the last control mechanism, local tissue control and hormones. This is where the smooth muscle acts as its own brain, particularly regarding blood flow.
This is absolutely critical for matching blood supply to metabolic demand. The tiny arterioles that feed your tissues have very little nerve supply. They don't wait for a signal from the brain. They are highly responsive to the local chemical environment they are sitting in.
What are they tasting for in that environment?
They are tasting for signs of need. If a tissue is working hard, say your quad muscle when you're running, it rapidly uses up oxygen. A lack of oxygen in the local tissue fluid is a powerful signal for the smooth muscle in the arterial wall to relax. That's vasodilation.
So more blood carrying more oxygen flows in to meet the demand.
Exactly. It's a perfect negative feedback loop. If the tissue produces a lot of waste products from metabolism, like excess carbon dioxide or lactic acid, or an increase in hydrogen ions, which means it's becoming more acidic, all of these are also potent vasodilators. They make the smooth muscle relax.
It's the muscle essentially saying, the tissue here is stressing out, open the floodgates and give it what it needs.
That's precisely what's happening. Adenosine, which is released from breaking down ATP, is another big one. Even increased body temperature in a local area will cause dilation. It's a beautifully intelligent local control system.
And then there are the hormones. We listed a whole bunch earlier. Norepinephrine, epinephrine, angiotensin II, vasopressin, oxytocin, serotonin, histamine.
The list is very long. The key takeaway is that the entire body uses the bloodstream as a communication network. These hormones circulate and can act on smooth muscle cells all over the body, provided those cells have the correct receptor.
And sometimes these hormones don't open ion channels directly, right? They can use second messengers.
Correct. This is a more indirect pathway. Sometimes a hormone binds to its receptor, and instead of that receptor being an ion channel itself, it activates an enzyme on the inner surface of the cell membrane, an enzyme like adenylate cyclase or guanylate cyclase,
Which in turn produces
CMP or CGMP. These molecules are called second messengers. The hormone was the first messenger. These guys go and do the work inside the cell, and in smooth muscle, their effect is often inhibitory.
Now they inhibit.
They often go on to phosphorylate and activate other enzymes, specifically pumps that actively pump calcium out of the cell or back into the SR.
So they lower the intracellular calcium levels, causing relaxation.
Yes. This is the mechanism by which many important vasodilator drugs and even asthma medications work. They increase these second messengers to force the smooth muscle to relax, which opens up the airways or the blood vessels.
Wow. We have covered a massive amount of ground, from the microscopic side polar heads and the dense bodies, all the way up to systemic hormonal control and local chemical feedback.
It's an incredibly complex system, but when you step back and you put all the pieces together, you really start to see a theme emerging.
Versatility.
Versatility and resilience, exactly. Skeletal muscle is a high-performance specialist. It has one job, move bones, and it does it very fast and very powerfully. Smooth muscle is the ultimate generalist. It can be triggered by nerves, by hormones, by physical stretch, or by local chemistry. It can hold tension for days without eating up your energy reserves. It can collapse down to almost nothing and expand to huge volumes.
It is the absolute unsung hero of our physiology. We glorify the bench press and the marathon time, but the smooth muscle is quietly, efficiently keeping the entire machine running in the background.
It allows us to have an internal life. It automates all the critical survival processes so that our conscious brain is free to do other things, like worry about the bench press.
Here is a provocative thought to leave you with today. We like to think we are in charge. I decided to eat, I decided to run, I'm controlling my body. But right now, inside of you, there's a vast, distributed chemical intelligence that is monitoring your oxygen levels, adjusting your blood pressure second by second, pacing your digestion, and managing your waste, all without your permission. and entirely without your awareness.
We are, in a very real sense, just hosts to these incredibly sophisticated autonomous systems.
And my final question for you, the listener, to mull over is this. If this entire system, this engine of your survival, is so heavily and directly dependent on extracellular calcium, on that slow diffusion from the outside in, how does that change the way you look at that glass of water you drink or that meal you eat? You aren't just building bones or fueling your workout. You are providing the literal electrical spark for the engine of your interior world.
It's a powerful connection to make between what you consume and how your body just works.
Thanks for diving deep with us. We'll see you on the next one.